Hello once again, and let's continue with our chapter on enzymes. Okay, so in the previous lesson, we have learned about denaturation and also about how temperature can affect enzyme action. So just want to quickly run through this investigation with you. Just think about it in our mind. And so what is this experiment about? Okay, we have test tubes A, B, C, D. We fill them with starch solution. Then we have we dilute it with some water. Okay, never mind. Basically, A, B, and C, right? They contain the starch solution with amylase solution. So what do you think will happen to the starch? Okay, we know that starch is gonna be digested by the amylase into maltose. But is it possible to happen if it's at 0 degrees Celsius? Is it possible for it to happen if it's at 37? Is it possible for it to happen if it's at 100 degrees? What will happen to the amylase at 0? Right? It's probably going to be inactive. What's going to happen to the amylase at 100? It's going to be denatured because it's too hot. Okay, this one is just a control. A control is just to ensure that your experiment is fair and reliable. Okay, and then blah blah blah. Let's just watch the video for this, and then we can see how actually it works. Okay, so it will look something like this, right? You have your starch solution with your amylase, and then you pour the amylase into the starch, and after that you test it with iodine. Okay, now remember, when you use iodine test, it will turn, right? The solution will turn from brown, right, to blue-black if there is starch present, and it will remain brown if there is no starch. Okay, so let's watch this video.
Label 6 test tubes A to F. Add 1 cubic centimeter of amylase solution to each tube. Label another 6 test tubes 1 to 6. Add 5 cubic centimeters of starch solution to each tube. Group tube A with tube 1. Tube B with tube 2, and so on. Put the test tubes into beakers of water at different temperatures. Leave the tubes in the beakers for 10 minutes.
Okay, why is it important to incubate the two solutions before mixing them together? This is so that you can ensure that the liquid that's inside the test tube, both the test tube, are at the correct temperature, at the same temperature, be it 10 or 20 degrees Celsius, etc. Okay, so why was that at 10 minutes? That was to give the enzyme sufficient time, right, to work on the substrate and hopefully turn it into the products, right, which would be maltose.
For each beaker, start the reaction by pouring the starch solution into the amylase solution. Shake the mixture well. Start the stopwatch immediately and record the time as 0. At 2-minute intervals, transfer a drop of each reaction mixture to an iodine drop on the stop plate with a clean dropper.
Okay, so here they are doing what it calls an interval. That means that you take a bit of the liquid out and you test it every few minutes.
Stop when the iodine drop remains brown.
Okay, so you see, so at different intervals, so like this one, it remained black all the way. Okay, why did it remain black all the way? Because there was no enzyme reaction, there's no enzyme activity. So that means that there's the starch was still there all the time, right? No matter how long it took for the whole experiment to take place, at the end of the day, there was still starch in that test tube. Okay, but as compared to this, we start to see that after some time at the bottom, like let's say
this is the time at which all the starch in the mixture is broken down. Record the time.
Okay, so this one. So you see, at 0 degrees Celsius, the blue-black color does not disappear at all, no matter how long, right? Because the enzymes are inactive. At 20, it takes 6 minutes, right, for the blue-black color to disappear. That means that it remains brown. Okay, this is because it takes 6 minutes for the enzyme to completely digest all the starch into maltose. At 40, it takes 4 minutes, and at 60, it takes 2 minutes. Okay, so this probably indicates that this particular amylase is working best at 60 degrees Celsius. Okay, although it's a bit high, might not be human amylase, might be some other amylase. But anyway, the shorter the time, right, the higher the activity. Okay, because it's very fast. However, at 60, there is a chance that it might become denatured after some time. Anyway, talking about 80, 100, obviously, the blue-black color did not disappear because the enzymes have been denatured. And so, they will no longer be functional. Okay, they cannot bind to form the enzyme substrate complex anymore.
Present the results in a table.
Okay, so what we see is that enzyme activity does change with temperature. There's going to be an optimum temperature for the enzyme where the time taken for it to completely digest the starch is shortest, which means that the rate of activity is the highest. Okay, so let's talk about the graph. Okay, so when it comes to graph for temperature, you need to realize how important it is to use the data in the graph. Now there will be some data, let's say for example, there's some data about the temperature and some data about the enzyme activity. Okay, this is not the best. Okay, let's search for another one. Temperature, graph, enzyme. Okay, let's see if we can find one good one. Let's just see this one. Okay, it doesn't really matter what enzyme this is, but we just use it as an example. Okay, so it's a bit blur, but let's look at the first chart here. We see that there's this enzyme activity from 20 to 100. Okay, there's usually no units. I just call arbitrary units for enzyme activity. Okay, so what we can say, how will we describe this graph? How to use this graph? Okay, we will start by saying at 10 degrees Celsius, the enzymes are inactive, and the rate enzyme activity is at about, I'll say about 3 or 4. Okay, so as the temperature increases, the effective collision between the substrate and the enzyme increases, and there's more enzyme substrate complex being formed. And so the optimum temperature for this enzyme is about, okay, say 33 or so. I think, okay, use a ruler, measure down. Okay, so the optimum temperature is 33 degrees Celsius, where the enzyme activity is at about 95, 96, around there. Okay, and this is the temperature at which the enzyme's activity is the highest. Above the optimum temperature of 33, the enzyme activity starts to decrease because the enzymes are starting to become denatured. At 60, at 58 degrees Celsius, the enzymes are completely denatured and there is no more enzyme activity. Okay, so that's how you use a graph to explain. You make reference to the numbers on the x-axis and on the y-axis if there are numbers. Okay, if there are no numbers, then you can just make a general statement. Okay, you try to do this for the second graph over here. Okay, so you say enzyme activity, temperature, denature, all that kind of stuff. You try it yourself. Okay, now I will continue. You can pause the video, but I will continue, and I'll talk about how enzymes are being affected by pH. Okay, just a quick side note. You should realize that our body has different areas or different organs with specific pH. For example, our mouth, saliva, right, the pH is 7. So the amylase in our mouth tends to work at optimum pH at 7. There is the stomach, which has a pH of 2, because the stomach produces hydrochloric acid. And so the proteases that function in the stomach, which is actually called your pepsin, is gonna have the optimum pH of 2. And there's also proteases that function in the small intestine. Now the small intestine is a slightly higher pH, alkaline pH of 8. The reason is because the juices, the things like the bile that's produced from the liver, that's secreted into the small intestine, and the pancreatic juice, small intestinal juice, these juices, they are actually slightly alkaline. Why is it alkaline? They are there to neutralize that hydrochloric acid that came from the stomach. Remember, the food is coming from the stomach to the small intestine, right? So it comes as a very acidic like substance. So the small intestine is going to produce some alkaline liquids to neutralize the acidity. Okay, so enzymes are proteins, so they'll be affected by temperature and pH. Okay, let's talk about pH. What is pH? It's just a scale, right? On the scale of 0 to 14, 0 being super acidic, strong acids, and 14 being strong alkali, and 7 being neutral. Okay, so enzymes are affected by the acidity or alkalinity of the solutions in which they act. Some work best in acidic solutions, for example, the protease in the stomach, whereas others require alkaline solution, for example, the intestinal enzymes. When enzymes are placed in pH conditions that vary from the optimum, they may start to denature. Extreme changes in the acidity or alkalinity of the solutions will denature the enzymes. So in this figure, point M indicates the maximum activity of amylase at pH of about 7. It's the optimum pH. As the solution becomes acidic, which is 5 and below, or if it becomes alkaline, which is like around above this, right, above 7, then the activity starts to decrease. So at pH 4 or above pH 9, the enzyme is completely denatured. A little bit funny, why they say 9 when it's clearly 8 here. Okay, I don't know. Okay, anyway, how would I say this? Okay, okay, I want to talk about this one, protease in the small intestine. Okay, so I will start by stating the optimum pH for this enzyme first. So I'll say the optimum pH for protease in the small intestine is pH 8. At extremely low pH, such as 5.5, or extremely high pH, such as 9.5, I don't think this should be 9.5, I think so, the pH will cause the enzyme to become denatured and the rate of enzyme activity will decrease. Okay, so a bit simpler as compared to the temperature one. Okay, you can try one more time over here. Okay, so you can try this for pepsin. Pepsin is the protease that's found inside your stomach. So what is the optimum pH of pepsin? You can say is, okay, actually it's 2 lah. Okay, so let's say the optimum pH of pepsin is 2, and when it goes to 0, which is too extremely acidic, which is impossible in our stomach never get to 0, but if it does go in the lab or something, it might be denatured. And if it becomes at pH 5 point something, 5.8 something, then the enzyme is too alkaline for the enzyme, and it also becomes denatured. Okay, so just some cool stuff. These COVID vaccines, right, which we took recently, they actually have to be stored at very, very cold temperatures, negative 70 degrees Celsius. The reason is because they are trying to stop the enzymes from working, right? So there might be some enzymatic reactions that break down the mRNA. The mRNA is a kind of genetic material that is important for us in the vaccine, and so it needs to be frozen. Okay, so these vaccines are actually frozen before they are administered to us. So only when we need it, then they take it out from the fridge, they thaw it out, everything, get it ready, and then they can administer it. They can't put it back, by the way. Once it's open, then they have to use it all the way or throw it away. Okay, so let's talk about these views from these people. B says, if a shirt is made from silk and silk is a protein, can the shirt be digested by enzymes? Let's find out. A says, I don't think that can happen. Many people wear silk. Moreover, silk is not a nutrient. How can it be digested? C says, I think it can happen, especially if the right enzymes are used. Since silk is a protein, it can be digested by protease. I'm not going to wear any silk. Okay, so the answer is that C is semi-correct, okay, but the likelihood of you encountering protease out in the wild, like suddenly the wind blow protease onto your shirt, is very unlikely. But yes, silk can be digested by specific enzymes, right, that can target the silk. Okay. Okay, this one, you read it and then you try to imagine the scenario. Okay, imagine the scenario ahead. This one is basically like trying to test the optimum pH of the pepsin. Okay, so you can think about it as a bit similar to just now what we saw in the iodine experiment, except that was about temperature, right? This one is about pH. So what do we have here? Right, this hydrochloric acid, the 10 drops of hydrochloric acid will make the pH acidic, and this sodium carbonate will make the pH more alkaline, and then distilled water is just neutral. Okay, so which do you think the protease will be most active in? Now remember, this protease comes from the stomach, right? So this protease most likely will be working best in the acidic environment. So why is B there for though? Why A got protease, then B got distilled water? What do you think that's for? And this is what you call as a control experiment or control setup. So you want to ensure that the reaction is caused by the enzyme and not anything else. So you must rule out any other possibilities. So that's why there's this thing called control of using water. Okay, so do this practice question yourself. Name the 2 properties of enzyme. State 2 other features that are not in the picture. Okay, and then tell me what is denaturation also. Okay, you see this theory book? It's time to do your theory also. Okay. Alright, let's finish up the chapter with the mind map. So, enzymes. Enzymes are biological catalysts. Do you remember the definition of the enzyme? Go and memorize it. Okay, then the enzyme has a activation, lowers the activation energy over here. Okay, so let's start from here. What are the functions of the enzyme? The function of enzymes are to digest large food molecules into smaller food molecules, break down food substances in cells to release energy, build up or synthesize complex substances, or break down toxic substances in the cell, right? The example is the catalase. Okay, then the characteristic of the enzyme is, firstly, it speeds up chemical reactions without being chemically changed. It is required in small amounts. It's highly specific. It works best at an optimal temperature and pH. And okay, most enzymes actually catalyze a reversible reaction. That means that they can actually like from protein can become amino acid can become back to protein again. Okay, it's called a reversible reaction. Some of them are actually irreversible. Okay, then mode of action. How does it work? The enzyme provides an alternative pathway with lower activation energy required for a reaction, and interacts with the substrate according to the lock and key hypothesis to form a enzyme substrate complex. What are some reactions? Carbohydrates digest carbohydrates, protease digest proteins, lipase digest fats, known as lipids. Okay, these characteristics and mode of action are affected by the changes in the temperature of pH. So increasing the temperature increases the rate of enzyme reaction until the optimum temperature is reached. Increasing or decreasing the pH from the optimum decreases enzyme activity. Enzyme denaturation takes place when there are extreme changes in the pH and at a temperature above the optimum. Okay, so be aware of how to draw these 2 graphs. They look similar but not the same. Okay, so for the temperature graph, it's a bit like a roller coaster, right? You go up slowly, slower, and then you drop down fast. Whereas for the pH graph, it's a bit more like a normal mountain shape. Okay, so it's more balanced and more equal looking. So please draw them correctly and not just anyhow draw if you have to draw. Okay, and with that, I want you to do your review questions. Okay, and then we'll go through the answers in class. Okay, so that's the end of chapter 4. Alright, do take note, chapter 4 and chapter 3 are actually usually combined together. Okay, so in your O level or N level exam, actually this is considered as one chapter only, so you must study them together. Okay, all right, and take care. Bye bye. See you in school.